Chapter 5, Part 1, The Structure and Function of Plasma Membranes. Plasma membranes, also called cytoplasmic or cell membranes, are fluid mosaics. We have biochemicals that are arranged closely together, but they're not all covalently bound or bonded together. It's a very unique structure that's associated or held together often by weak interactions as well as strong interactions. Here we see a typical example of a fluid mosaic biological membrane called the plasma membrane. Biological membranes are other kinds of membrane structures as well as the plasma membrane. So biological membranes tend to have this type of two-layer system or organization with other types of structures either embedded through the layers of the structure or even attached to the surface, both on the outer and inner surface. This original fluid mosaic model was proposed in 1972 by Singer and Nicholson and took a little bit of research to actually confirm, yes indeed, biological membranes tend to have this type of organization. Plasma membrane function includes defining the outer borders between other cells and organelles, managing what enters and exits the cell, receiving external signals, initiating a cellular response that then is transmitted back out of the cell, and allowing a place for one cell to attach to another, allowing for the formation of tissues. The main building block of any biological membrane is a phospholipid. Go back to the chapter on macromolecules and chemistry to review the basic structure of a phospholipid. It is the first major component, formed of two fatty acid tails that are nonpolar or hydrophobic, and a hydrophilic head that's composed of a particular structure called a phosphate-rich head or polar group. The polar head, or the phosphohead of the phospholipid, is going to be the unique component that allows for the easy organization of a biological membrane. Each fatty acid, which is the nonpolar component, remember the two fatty acid tails, can either be saturated or unsaturated. If unsaturated, with at least one double bond that forms a kink or a bend in the fatty acid tail, allows for some flexibility. If the fatty acid tail is saturated, then we're going to see that the tails can be packed more tightly together. Phospholipids are arranged with the polar heads, illustrated as white spheres in these models in the illustrations, outward towards the water-rich environment. Polar heads are hydrophilic. They're going to be attracted to water. The hydrophobic tails, which are illustrated in tan, or this golden color, are hydrophobic. They're going to steer away from water and point inward. Now, the phospholipids can form double layers with a layer of hydrophilic heads on the inside and a layer of hydrophilic heads on the outside. That's the typical formation of a phospholipid bilayer we see in biological membranes, illustrated at the bottom. Or we can even have those as smaller packages that we call liposomes. Liposomes, again, will have a hydrophilic center where the heads of the phospholipid layers are pointing toward the center, and the phospholipid heads that are hydrophilic pointing on the outside, and the bilayer construction, the hydrophobic tails associating with one another in the middle. Some medications can be packaged in liposomes for delivery systems to our cells and successfully work that way. We can also have monolayers, a single layer of phospholipids with the hydrophobic tails pointing toward the center and the hydrophilic heads on the outside surface. Whenever we're packaging lipid-rich chemicals, for delivery systems, we might use micelles. So if it's a lipid-rich, non-polar chemical that we want to package as a medication, then we'd use a monolayer or micelle type of delivery system. Proteins are the second major component of membranes. They can function in many types of ways: transporters, receptors, enzymes, adhesion or attachment structures from one cell to another. And we have two general categories of proteins that are found associated with membranes. Some of them are integral proteins. They're actually what we call integrated completely throughout the bilayer structure. We can also have peripheral proteins that are only found on the surface of the cell. Integral proteins are also called transmembrane, meaning they go trans. They go all the way through from one edge of the membrane to the next, inner and outer. The portion that's embedded in the membrane are composed of both hydrophobic and hydrophilic amino acids. Generally, the center component of the transmembrane protein is hydrophilic, and the outer surface that's attached to or more associated with the fatty acid tails of the phospholipid layer are going to be hydrophobic. The yellow band in this illustration is supposed to show you the phospholipids. So this yellow band is the phospholipid layer, and the nonpolar tails are in the middle or spanning between one surface and the next, and then the transmembrane protein is embedded between the polar heads, spanning the width of the membrane, and it's the hydrophobic portion on the outer surface of this transmembrane protein embedded in the membrane that keeps everything stable. The third major component of a membrane are the oligosaccharides. Oligo means scant. So oligosaccharides are just small carbohydrates, small saccharide arrangements. Remember, saccharide means sugar. A monosaccharide would be a simple sugar, like glucose, galactose, or fructose. An oligosaccharide means a short chain of sugars. They can be located on the exterior surface. They can be found attached to proteins and lipids. We can have sugars that are attached to proteins. We call those glycoproteins. We can have sugars attached to lipids. We call those glycolipids. Notice that the glycolipids and glycoproteins often have the sugars sticking out from the surface of the membrane. These are going to function for attachment as well as cell-to-cell recognition and interaction. An example of a receptor or type of protein or glycoprotein on the surface of a cell is the CD4 glycoprotein of what we call T lymphocytes or T cells. That glycoprotein that's recognized or called a CD4 glycoprotein is the doorway, if you will, or the attachment site for the human immunodeficiency virus. It's how this virus gains entry into our cells and results in an HIV infection. Membrane fluidity and flexibility is very important. The membrane needs to be fluid and flexible, but not to the point that the structure completely breaks down or falls apart. So the types of fatty acids are very important. If we want a really rigid membrane, then we want saturated fatty acids. Saturated fatty acids can be packed very closely together. If we need a more flexible membrane, then some of the fatty acids must be unsaturated, because all of those double bonds resulting in the little kinks of the fatty acid tails keep them separated a little bit more. For colder temperatures, we want the membrane to be more fluid. For hotter temperatures, we want the membrane to be more packed together or solid. And in fact, the composition of your fatty acids in your membranes does change from one season to the next. particularly if you spend a lot of time indoors and outdoors, and you go especially between different temperatures and work inside or outside more. Cholesterol between the phospholipid molecules helps to keep membranes fluid. The cholesterol will be, in fact, sort of wedged in between the phospholipids.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
There are various mechanisms are used to bring materials inside and deposit materials outside of a cell. Endocytosis, endo means within, cytosis is a mechanism of the cell, brings materials in. We have 3 types of endocytosis. Phagocytosis is a specialized type of endocytosis that brings particles inside of a cell. Pinocytosis means sipping, bringing liquids inside of a cell. And receptor-mediated endocytosis is a type of selective transport. The illustration here shows the generalized type of endocytosis, bringing materials in through invagination or the blubbing into the cell to form a little vesicle that then can be used to transport materials throughout the cell. If it's a particle, we can see in the left-hand illustration here, where the whole particle is brought inside of the cell. In this example is a food vacuole. The whole structure is a solid particle, is surrounded by cytoplasmic membrane, invaginated, surrounds the stretching of the plasma membrane surrounds the particle, captures it in what's called a phagosome, which then can be further broken down or further modified to bring that food in. For pinocytosis, we're bringing in liquids from the outside surface. And for receptor-mediated, we have selective attachment of certain types of materials that the cell needs that will be captured by those structures that we call receptors. You can see them in green. They're going to capture specific kinds of nutrients or specific kinds of chemicals. Again, we have a stretching of the plasma membrane surrounding those items and bringing them into the cell in a vesicle. Here we see an amoeba encountering another protist called stentor. Now you would think that the amoeba would be the food for the stentor. So the amoeba is on the top, and it moves by a type of mobility structure called pseudopodia, which results in the stretching and the elongation of the plasma membrane of the amoeba. Amoeba are single-celled protists. The stentor is also a single-celled organism. It has a mouth type of structure and opening that's surrounded by cilia that allow the stentor to bring food and nutrients into its cell. But look at what happens when this amoeba, stretching around and enlarging and flexing its plasma membrane, comes in contact with these stentor cells. The pseudopodia, or the false feet, are stretching around the stentor, and slowly but surely this amoeba is going to capture and eventually totally engulf the stentor as a source of food. This is a very specialized mechanism or type of phagocytosis. It takes the single-celled amoeba a little bit of time to engulf and ingest the stentor, but eventually it will successfully completely surround this, what we call ciliated protist, the stentor, and consume it as a source of food. This motion picture, or these images, are in real time. So you can see, slowly but surely, this amoeba is going to finish its task of getting a really good meal of this single-celled protist, the stentor. Exocytosis is just sort of the opposite. Once we've formed a vesicle with some kind of substance that has to fuse with the plasma membrane, it's transported from the center of the cell to the exterior surface. It fuses with the plasma membrane, and the contents are either dumped out, or perhaps, if they are embedded in the vesicle membrane itself, the membrane of the vesicle fuses with the plasma membrane and adds material to the cytoplasmic membrane. We find that proteins, especially glycoproteins or lipids like glycolipids, actually form rafts on the surface of the plasma membrane. They're called rafts because the chemicals that were fused together in the membrane itself float around just like a little raft on the surface of the water.